All right. By way of review, I think you all know that this is a general model for a eukaryotic mRNA. The mRNA has a cap at the 5' end and a poly A tail at the 3' end. It has an open translational reading frame, which is the part of it that encodes protein. The open reading frame begins usually with an AUG, translation initiation codon for methionine, and ends with a codon, one of 3 codons, UAA, UGA, UAG, that do not encode an amino acid and signal translation termination. Also, most mRNAs in mammals and a few notable ribosomal protein RNAs in yeast and a few others undergo splicing. This line represents the exon-exon junction that resides within spliced mRNAs. There are exceptions to this structure. For example, cell cycle regulated histone mRNAs lack a poly A tail. And these cell cycle regulated histone mRNAs, as well as mRNA for the proto-oncogene protein cellular June, doesn't derive from genes that have introns. These mRNAs do not undergo splicing. All cellular mRNAs are eventually degraded, and decay is a highly regulated process. mRNA decay is often coordinated with transcription. And when you think about it, it makes sense, because if it's time for a cell to eliminate the production of a particular protein, yes, it can downregulate the transcription of the gene that encodes that protein, but it would be more efficient if it also post-transcriptionally degraded the mRNA. So it reduces a rate of, I don't know if I just said degrade transcription, did I? It reduces the rate of transcription and degrades the product mRNA. mRNA half-life, and I should define that, half-life is the time it takes for half of existing mRNA molecules in a cell to be lost, to be degraded. Half-life is controlled by specific interactions between cis-acting elements in the mRNA and the proteins that bind these elements. Some of the regulators are general regulators. So to go back to this slide, proteins that bind an mRNA cap are stabilizing because they protect the 5' end of the mRNA from nucleases. Same with proteins that bind the poly A tail. Poly A binding proteins are generally stabilizing in the sense they physically protect the poly A tail from deadenylases, nucleases that degrade poly A. Oftentimes, regulatory elements in the 3' and translator region control mRNA half-life. We'll talk about a number of these, like AU-rich elements or AREs, which are generally destabilizing. The process of translation itself through the open translational reading frame can influence mRNA stability in the sense that the ribosomes can remove a protein that either stabilizes or destabilizes the mRNA. And then, as you'll see from the second talk that I give, I think it's tomorrow, that the process of translation can be required for mRNA decay. And the examples that I'll talk about are nonsense-mediated mRNA decay and Stauffen-mediated mRNA decay, both of which require translation termination before the mRNA can be degraded by those processes. All right. Eukaryotic mRNA half-lives can vary from minutes to hours. And examples include the beta-globin mRNA, which has a relatively long half-life of 17 hours. Beta-globin is a constituent like alpha-globin with to make up hemoglobin. And the proto-oncogene C-June mRNA has a very short half-life of 15 to 20 minutes. Generally, organisms that have a rapid rate of cell division have mRNAs with shorter half-lives than cells that have longer rates of division. And finally, changes in mRNA decay can allow for fine-tuning of gene expression and can occur in response to developmental or environmental stimuli such as nutrients, cytokines, hormones, and temperature shifts, as well as stressors such as viral infection, hypoxia, or tissue injury. An example is illustrated by casein mRNA half-life. This increases from 5.4 hours to a relatively long 92 hours when mammary tissues are exposed to the peptide hormone prolactin. With this increase in half-life is a concomitant increase in transcription. Transcription rates are increased about 2 to 4-fold, and both the increase in transcription and increase in the half-life of the mRNA occurs within 1 hour of exposure of the cells to prolactin. Another example of regulated half-life derives from histone mRNA, the cell cycle regulated histone mRNAs. They are degraded at the end of S phase of the cell cycle. When DNA synthesis is over, there's no more need for new histone biosynthesis. And what happens is the half-life of histone mRNA decreases from about 45 to 60 minutes to 10 minutes at the end of S phase. And concomitantly, there's a downregulation of histone gene transcription. This slide illustrates that a subtle change in mRNA half-life can manifest a significant change in mRNA abundance. The examples are of an mRNA that has a 40-minute half-life and another mRNA that has a 10-minute half-life. And provided the transcription of the genes that encode these mRNAs stops at time 0, there is a significant difference in the steady-state level of these mRNAs by 2 hours. By 2 hours, the mRNA with a longer half-life is reduced to about 25% of its initial level, whereas the mRNA with a shorter half-life is reduced to about 0.4% of its initial level. Very efficient way to downregulate gene expression by concomitantly blocking transcription and increasing the decreasing the half-life of the mRNA encoded by the gene. Generally, there are 3 cytoplasmic mRNA decay pathways, 2 of which depend upon deadenylation, and 1 of which does not. Deadenylation initially occurs by a complex of proteins called PAN2 and PAN3. PAN stands for polyaribonuclease. These 2 take what is a newly synthesized cytoplasmic mRNA that generally has a poly-A tail of about 200 to 250 A's and reduce that tail to about 100 and 110 A's. This complex is activated by poly-A binding proteins. So this complex likes a long poly-A tail as a substrate. In fact, PAN3 physically directly interacts with poly-A binding protein C1, C1 being the major cytoplasmic poly-A binding protein in mammals. It's called PAB in yeast. Then, after the tail is shortened to about 100 nucleotides, another deadenylase takes its place. This doesn't like long tails as much. It's a complex that's abbreviated CCR4 NOT, both of which can have deadenylating activities. And actually, there are many types of CCR4s and NOT proteins that can constitute the complex. In fact, people have shown that PAN3, PAN2, and CCR4 NOT can exist in a supracomplex. So these 2 can recruit this with the poly-A tail is long and there's a lot of poly-A binding protein. CCR4 NOT reduces the poly-A tail to an oligo-A length. It's 10 nucleotides or less, after which 2 things can happen, and often both happen. One is decay of the body of the transcript in the 3' to 5' direction by a complex called the exosome. And then the other is decapping. DCP2 is the decapping enzyme, and it's activated by DCP1 as well as a number of activators that we'll discuss. After decapping occurs, then the body of the transcript can be degraded in the 5' to 3' direction by the exonuclease called XRN1. So you might be sitting there saying, okay, I understand when the poly-A tail is shortened, the exosome can then degrade what is an unstructured little oligo-A tail and the rest of the transcript, but how does removal of the poly A tail to an oligo A form activate decapping? You might be saying, I don't understand that. And that can partially be explained by what's called the closed loop model for mRNAs. So this pertains to yeast as well as mammals. And the idea is that the mRNA is circularized, kind of, transiently. And the way it can be circularized is via a platform protein, which is a translation initiation factor. So I'm showing you the steady state translation initiation complex. This is a complex that the bulk of cytoplasmic mRNAs is in. This eukaryotic initiation factor 4G, or EIF4G, serves as a platform to physically connect poly A binding protein C1 and the cap binding protein called eukaryotic initiation factor 4E. Once the poly A tail is shortened to an oligo A form, then PABC1 cannot bind to it anymore. It cannot bind to an A tract that's less than 2 nucleotides. So this complex becomes undone, and this destabilizes 4E from binding to the cap. And remember I said that cap binding proteins stabilize mRNAs by physically protecting the 5' end. So without EIF4E bound to the cap, then the mRNA is accessible to decapping. All right, so we've talked about the two deadenylation dependent pathways. There are endonucleolytic pathways as well, and I've listed a few endonucleases. PMR1 is polysomal mRNA nuclease, and it's involved in degrading mRNAs that are responsive to estrogen. This endonuclease is involved in the innate immune response. It helps degrade mRNAs so that once we raise an immune response, it just doesn't go haywire, it comes to an end. The inositol responsive protein 1 is involved in the unfolded protein response, and when proteins become unfolded, it's important to stop mRNAs entering the secretory pathway. And so this protease, nuclease, endonuclease, degrades mRNAs that encode secreted proteins. And we will talk a little bit about the endonuclease SMUG6, which is involved in nonsense-mediated mRNA decay. After the endonucleolytic cleavage, which is shown by this Pac-Man here, then the 3' cleavage product can be degraded from the 5' end by XRN1. There's no cap here. There's nothing to prevent it from degrading the RNA. And the 5' cleavage product has nothing in the way to prevent the exosome from degrading the 5' endonucleolytic cleavage process by the 3' to 5' exonuclease, the exosome. I had mentioned that the process of translation itself can actually influence mRNA half-life, and the example that I'd like to give is for CFOS mRNA. This is an mRNA that encodes a proto-oncogene protein, so it inherently has a short half-life, and bottom line is when translation of CFOS open reading frame occurs, then the mRNA kind of undergoes suicide. It's degraded. These experiments were done by Enbin Shui's lab, and they were reported in 2004, and I'll just describe some of them. So in these experiments, Enbin's lab transiently transfected NIH3T3 cells, which are an adherent cultured fibroblast cell line from mouse, with one of two plasmids that produced one of the two mRNAs shown here. These are hybrid mRNAs. B stands for beta globin, F stands for CFOS, and B stands for beta globin again. So these mRNAs, this one in particular, has the coding region from CFOS mRNA and the 5' and 3' untranslated regions from the beta globin mRNA. The only difference between this and this is that this hairpin-containing mRNA has a hairpin in the 5' untranslated region that inhibits translation because the 40S ribosome that recognizes the cap and then scans to find the translation initiation codon can't undo the hairpin. It doesn't have nearly the unwinding capability as the ADS functional ribosome. So this is a control for whether or not the decay process that this lab is going to study depends upon translation or not. And it does depend upon translation. So what they did is they introduced plasmids into cells transiently and then deprived the cells of serum. And what this did, because both of these mRNAs were produced from the CFOS promoter, was provided a transient burst in transcription, meaning transcription was activated, and by 30 minutes of serum addition to serum-deprived cells, the promoter was shut off. And during that shut-off period, remember, you really don't want ongoing confounding transcription to study mRNA half-life. They could measure the half-life of the mRNA that was synthesized in that burst of transcription. And what they did was they performed a northern blotting analysis. They're measuring mRNAs and a control alpha-globin GAPDH1 mRNA. But let's look at the experimental mRNA, the BFB mRNA, which is here. So as I said, the burst in transcription is maximal by 30 minutes after the addition of serum to serum-deprived cells. And you can see that not only does the amount of this mRNA decrease with time, but its size does as well. So what this is saying is the mRNA is concomitantly deadenylated and then degraded. Its body is degraded. And as a molecular weight standard, they took cells, isolated RNA, and treated that RNA with RNase H and oligo-DT. So oligo-DT is a DNA that anneals to the poly A tail of the RNA, and RNase H degrades the RNA strand of an RNA-DNA duplex. This is a control for a completely deadenylated RNA, and you can see that the RNA, in fact, undergoes deadenylation as well as decay of the body of the transcript. Whereas the mRNA that cannot be translated because it has the hairpin structure in the 5' translated region does not undergo significant decay. And this is just a polysome profile. This is a sucrose gradient, kind of like the glycerol gradients that John talked about, that separates RNPs, RNAs and associated proteins, based on size. And I won't go through the details, but all of this does is confirm that the hairpin does preclude translation because the mRNA is not associated with polysomes. And then what is the deadenylase that's degrading this mRNA? And they're performing the same kind of experiment, only they're using a beta-globin transcript that instead of having the entire open reading frame coming from CFOS's open reading frame, they're including just a little 87 nucleotide piece that includes the major coding region determinant, which is the region within the open reading frame of CFOS mRNA that they mapped to contain this destabilizing element that triggers deadenylation when ribosomes read through it. And in this experiment, they again performed northern blotting so they could analyze the consequence of expressing different variants of CCR4, which is a deadenylase, or PARN, which is another type of poly-A ribonuclease that I'm not going to discuss too much, or PAN2, either wild-type variants or mutant variants or an empty vector. So in the place of nothing being added, after serum is added to serum-deprived cells, so the transcription was turned on and then shut off by 30 minutes. This is the normal deadenylation and decay rate of the hybrid constructs, mRNA. And you can see that it's really not affected when CCR4 is overexpressed, but with a mutant CCR4, the decay rate is slowed. So this says CCR4 is an important deadenylase for the decay that's triggered by the major coding region determinant of CFOS mRNA. PARN's not involved, PAN's not really involved. And you're going to get these slides as a part of a disc, so you'll be able to review them as well as the initial paper if you're interested. This is just a Western blot showing that they indeed expressed the different deadenylases. And this is an immunoprecipitation where they showed that a V5 tagged CCR4 deadenylase, in fact, co-immunopurified with a protein called UNR that had an HA tag. The two proteins are present in a complex in cells. And nobody really understands how this happens. It looks like UNR can interact with the major coding region determinant and pull poly-A binding protein off the poly-A tail, and that makes it accessible to decay by CCR4. So that's the state of understanding this decay pathway right now. Do you have any questions? Okay. So just by way of review, how do mRNAs become deadenylated, undergo deadenylation? We've talked about the major coding region determinant in the CFOS mRNA, which is a labile mRNA that once translated gets degraded. It's a transforming protein. So a virus has assumed the CFOS gene as a viral FOS gene and helps promote cell transformation. It's a very dangerous message to be expressed promiscuously in cells because it will transform cells. And I would like to talk about AU-rich elements, or AREs, that are present, as far as I know, always in mRNA 3' UTRs if they are present in an mRNA, and they also trigger deadenylation. MicroRNAs, for those of you who study microRNAs, also trigger the deadenylation pathway as well as other pathways. All right, so what about AU-rich elements? These are generally destabilizing elements, and they were discovered a long time ago in the mid-'80s. It turns out that if one takes the beta-globin gene, which you know has a relatively long half-life, and insert into it the AU-rich element from the 3' untranslated region of a cytokine mRNA that encodes granulocyte, monocyte, colony-stimulating factor, then the mRNA of the hybrid resulting mRNA will be only 1 to 2 hours. But if the ARE, which is AU-rich, were interspersed with CG, so the experimentalists mutated the ARE, the half-life is normal. So one needs a wild-type ARE inserted into beta-globin mRNA to trigger ARE function and decrease in the half-life of the beta-globin mRNA. So how was the ARE discovered? Oftentimes, if one sees nucleotide similarity between species, that's an indication of a conserved function. And what Sean Kamen discovered back in 1986 was that the ARE sequence between mouse and human in GM-CSF mRNA had 93% identity, which was higher than the identity of their coding regions. The identity of their coding regions was only 65%. And there can be a tolerable mismatch in the coding region because of the wobble position of at the third nucleotide of every codon. So because of this high similarity, they concluded that there must be some functional significance to the AU-rich element in the 3' UTR. This was a time when people didn't really know what 3' UTRs did. And so to show function, they inserted the ARE, either the wild-type ARE, that's A, this is the DNA level, AT-rich, or the mutated ARE that I already talked about, the GC-rich, and looked to see what happened to the steady-state level of rabbit beta-globin gene. This is the wild-type rabbit beta-globin gene that has no insert, and its steady-state level is quite high. Really can't see any RNA when the ARE was inserted into the 3' UTR, but the mutated ARE didn't affect the abundance of mRNA. These are not half-life studies. They're steady-state levels that show the abundance of the mRNA, and one conclude that one reason why the mRNA may be reduced in abundance is because the mRNA is destabilized. But of course, more thorough experiments would have to look at other aspects of gene expression, like transcription, but because the only change in these cases was to the 3' UTR, they can conclude that this change was because of the ARE they inserted. Okay. And I just want to mention that I'm telling you the basics of the enzymatic machinery, but it's really complicated in terms of how mRNA decay is regulated in response to environmental stimuli and stressors. And this just shows a very small piece of how signal transduction can affect ARE binding proteins. So TTP, tris tetraproline, BRF1, butyrate response factor. This is a KH RNA splicing protein, actually, that also regulates mRNA half-life by binding to AREs, and ALF1, which is an AU-rich factor 1, all bind to the ARE and destabilize the mRNA that contains the ARE. And notice how they are negatively affected when they undergo phosphorylation by a number of signal transduction pathways. YB1 and HUR can bind AREs and stabilize the mRNA. And generally, the function of a destabilizing ARE binding protein can be inhibited either by a process that precludes the ARE binding protein from binding the ARE, or by inhibiting the interaction of the RNA-bound ARE binding protein to a degradative activity. Oftentimes, these activated ARE binding proteins can physically recruit deadenylating, decapping activities to an mRNA. And I should tell you that I just wrote a review that's coming out March 6th with Dan Schoenberg that goes into a lot of details about how mRNA decay is regulated. And there's my email address, so if you want a copy hot off the press before it's even available on PubMed, you can email me and I'll send it to you. Okay. So what? Oh, I'm sorry. You mean you want to write my email address down? First name Lynne _ last name Maquat @URMC. No one doing science has my last name, so all you have to do is Google Maquat and you'll find me. I am really easy to find. And then you'll get my email address. You'll get everything. All set? Okay. All right, so after deadenylation, one of two things can happen, and one of them is the body of the transcript can be degraded by the exosome. The exosome has a nuclear and a cytoplasmic existence. In the nucleus, it is involved in processing of particular RNAs, often in an endonucleolytic way, which we'll touch upon in a minute. It processes SNO RNAs and snRNAs, but we're not talking about that because we're talking about cytoplasmic mRNA decay, and in that case, the exosome is involved in degrading mRNAs in the cytoplasm. Here is a picture of the structure of the 9-nucleotide core exosome. So to put this in perspective, this is this shown at the side. So there's a 6-subunit base that consists of 3 heterodimers. So here's one heterodimer, here's another heterodimer, here's another heterodimer. And then on top of that is a trimer of the proteins that are in the boxes. So here's the 6-subunit core that consists of the 3 heterodimers, and on top of that is the 3-trimer cap. And where RNA goes is right through here. There's a core. This, even though people used to think it had nucleolytic activity, if you look at the domains that could be exonucleolytic, they're all mutated. So this inactive core can interact with either or both of these 2 proteins that do have exonucleolytic activity. RP6 in yeast is confined to the nucleus, but in mammals, the ortholog, which is called PMSCL100, because it was found, antibodies to it were found in patients with polymyositis. Polymyositis. I always put an extra syllable in it, thank you, from the MD. Polymyositis, scleroderma, overlap syndrome. In humans, this ortholog is in the nucleus and the cytoplasm. And of course, we're interested in cytoplasmic mRNA decay. It's a 3' to 5' exonuclease. And RRP4 is also a 3' to 5' exonuclease in the cytoplasm of both yeast and mammals. The exosome is interesting because it leaves behind about 7 to 9 nucleotides, which explains, it leaves behind something, or let me put it this way, it can thread 6 to 9 nucleotides. If there's a little piece of unstructured RNA that's smaller, it can't get to it. But this becomes important when one thinks about the oligo-A tail that's left over after deadenylation, because that will fit nicely into the exosome. So after the CCR4 not deadenylase leaves a 6 to 9 nucleotide piece of poly-A, oligo-A now, that can't bind poly-A binding protein, that fits very nicely in the exosome, which doesn't really use itself, it doesn't use the cleavage of ATP for energy. However, the exosome can function with auxiliary factors. So for example, because it doesn't have any unwinding capability itself, it can be recruited to oligoadenylated RNAs through the GTPase SCI7, and that interacts with a SCI complex of 3 proteins, one of which is a dead box helicase that you've already heard about from John, which helps unwind the RNA and allow the exosome to proceed degrading the RNA body. We've already talked about how ARE-mediated decay can be promoted by the exosome because the ARE binding protein, like tristetraproline, will recruit the exosome to the mRNA. And then there's an interesting quality control mechanism called nonstop decay. I don't know if you've heard of this. So what happens if you have an mRNA that underwent polyadenylation in the wrong site, like within the coding region? Or what happens if you have an mRNA that doesn't have any termination codon because the termination codon was mutated and there's no termination codon downstream? How can the cell get rid of that? Well, the cell gets rid of that RNA by having SCI7, which is the GTPase that structurally mimics the translation termination factor, release factor 1, also elongation factor 1, and fits into the MTA site of the ribosome that's run all the way to the end of the RNA and then stuck. So you don't want to get your ribosome stuck on an RNA because then you're going to downregulate protein synthesis depending upon the abundance of this. And so SCI7 will recruit the exosome and get rid of these junk RNAs, these RNAs that were processed improperly or have mutations that make them non-productive. There was a lot of brouhaha a couple of years ago about the yeast RRP4 subunit of the exosome having not only 5' to 3' exonucleolytic activity, but endonucleolytic activity. It turns out that yeast don't require both RRP44 or RRP6. You need one or the other. They still don't do very well, but they're alive, and people thought, okay, there's functional redundancy. And nobody really knows what the endonucleolytic activity of RRP4 does. But in terms of humans, it doesn't really matter for mRNA decay. There are 2 orthologs of yeast RRP6 in humans called DIS3L1 and DIS. And it turns out DIS3L1 is the one that's in the cytoplasm, and it has no endonucleolytic activity. So in terms of humans, the exosome doesn't contribute to mRNA decay in the cytoplasm using endonucleolytic activity. But people realized that RRP4 could have endonucleolytic activity because it, like SMUG6, has a PIN domain. And this consists of 4 acidic residues, and I've told you what they are up here. What they do is they coordinate a metal iron that activates water for a nucleophilic attack on a phosphodiester bond of RNA. All right, we'll talk about SMUG6 when we talk about NMD next time. All right, so after deadenylation, you know the exosome can degrade the body of the transcript from the 3' end, and you know that it can also trigger decapping. And there are actually 3 different types of decapping activities. The best known are the scavenger decapping enzyme called DCPS. I don't even see it here, but it's DCPS. And then the major decapping activity that degrades after deadenylation called DCP2, oh there it is, DCPS, DCP2 and DCP1. DCP2 has the enzymatic activity, this is auxiliary, and this will decap the oligo-A form of an mRNA by generating M7D GTP and a monophosphate N that will then be degraded by the 5' to 3' exonuclease XRN1. DCPS is a scavenging enzyme. So what happens if the mRNA is primarily degraded by the exosome? There will be, remember, the exosome kind of stops after 10 nucleotides, so how the heck do you get rid of this little piece? Well, DCPS won't touch this because the body of the transcript is too long. It can't fit into this, but it will degrade the little piece that's left over from exosome-mediated decay to generate 7-methyl GMP and 2 5' phosphates that are okay substrates for the XRN 5' to 3' exonucleolytic activity, and then it also gets rid of this guy, or at least degrades this guy to 7-methyl GMP. There is a third activity called NUDIT16 that removes the cap of the U2 SNO RNA, and it's also degrading mRNAs. And you know, I don't even know if this is in the cytoplasm. It's just not very well characterized. So we can just know that there's another decapping activity, not really knowing what it does, what fraction of mRNA it degrades, and whether it's in the cytoplasm or not. All right. I just want to talk about activators of decapping. There's a whole bunch. And let's focus on, well, headless in mammals. It stabilizes the interaction between DCP2 and DCP1 to promote decapping. Then there's this ATP-dependent dead box helicase called DHH1 in yeast, or ROCK-P54 in humans, that together with PAT1, in humans, releases the cap binding complex from mRNAs. This interacts with an interesting ring of 7 proteins called LSM1 through 7, which specifically binds deadenylated RNAs, and it really likes oligo-U tracks. And so remember, there's deadenylation to an oligo-A form. That's what PAT likes to bind to, and these proteins, and they help to recruit the decapping activity to the mRNA. I hope we have time to talk about oligo-U tracks, but remember, histone mRNA doesn't have a poly-A tail. And what happens when it undergoes decay at the end of S phase of the cell cycle is it becomes oligo-uridynylated at the 3' end. This recruits the LSM1 complex and promotes decapping. And that unstructured U also is a nice template for the exosome. It's unstructured. The exosome, remember, likes unstructured pieces of RNA unless it has auxiliary proteins to help it unwind. And then this is an interesting protein that inhibits decapping. So Mike Collegian could never get DCP1 activity to be detectable in mammalian extracts. He could get DCPS, but not DCP2. And it turns out that extracts have an inhibitory protein. So just because you can't find an activity doesn't mean it's not there. It just might be inhibited by a protein. It's a very interesting protein, but it drove him crazy for a while until he figured it out. Okay, here are just summaries of how ARE-mediated decay can be activated. So this is tris-tetraproline. We've already talked about how it can recruit the exosome. It can also recruit the decapping activity. We've talked a little bit about how mRNAs that have either an oligo-A, or in the case of histone mRNAs, U-rich elements that recruit the LSM1 through 7 complex, and this together with the PAT protein can promote decapping. And then interestingly, again, Mike Collegian found that some mRNAs have a particular 10 nucleotide hairpin at their 5' end that specifically recruits DCP2. So these mRNAs are very efficiently undergoing DCP2-mediated decapping. So let's just review. As a general rule, mRNA-specific determinants of stability reside in 3' UTRs, as a general rule. They are often destabilizing, or if they are destabilizing, they often target the mRNA for decay from both ends. They can recruit deadenylating and exosome-mediated decay activities, as well as decapping activities and 5' to 3' exonucleolytic activities. And if they're stabilizing, they often prevent one or more nucleases from binding either directly, in the case of proteins that inhibit an endonuclease, or indirectly in the case of activities that degrade the mRNA from both ends. Let's talk about some examples, and I might finish early, which I suppose is good. Okay, so alpha globin mRNA, like beta globin mRNA, is a relatively long-lived mRNA. It's expressed normally in erythroid cells. There is a disease called hemoglobin constant spring. And in that disease, the translation termination codon is mutated so that it encodes an amino acid. And ribosomes, instead of terminating there, read into the 3' translator region. And for a long time, people couldn't understand why the resulting alpha globin mRNA was unstable. Well, it turns out the ribosomes, when they translate into the 3' UTR, translate through a Cu-rich element that recruits a complex called alpha CP. This is a complex that contains an HNRPK-like protein. And this is a stabilizing complex. It stabilizes alpha globin mRNA because alpha CP can interact directly with PABC1, which ties up the 3' end, so it's not accessible to deadenylating activities. When patients or people don't have a functional alpha CP complex, two things happen to degrade alpha globin mRNA. One is, without this interaction with PABC1, the poly-A tail can undergo deadenylation more rapidly than normal. And also, without protecting the CRE sequence, an endonuclease, shown by the scissors here, can cleave the RNA into two pieces. And we already know that the 5' cleavage product can be degraded by the exosome, and the 3' cleavage product can be degraded by XRN1, the 5' to 3' exonuclease. And also, the short oligo-A form that didn't undergo endonucleolytic cleavage can undergo exosome-mediated decay. So alpha globin mRNA undergoes a very rapid decay if this complex is either missing because of a mutation in the patient, or if there is a termination codon mutation that allows read-through, and in essence, extension of the open reading frame into the 3' translator region. And then the iron response element is pretty interesting. That resides in the 3' translator region of transferrin receptor mRNA. Iron is toxic in cells if the cells are flooded with free iron. So there is a way by which iron is chelated in cells so it can be used when it needs to be used, but otherwise isn't around floating free. And that chelating protein is ferritin. The transferrin receptor mRNA encodes a receptor that can respond to the intracellular iron concentration. When intracellular iron concentrations are low, then it behooves the cell to bring iron into the cell, so it behooves the cell to have more transferrin receptor, and therefore the mRNA should be stabilized. And that's what happens in the presence of intracellular iron. Then the iron response element binds the iron response element binding protein, and this precludes physically accessibility of the RNA to an endonuclease. And it's interesting because the IRE also exists in the 5' translator region of ferritin mRNA and regulates translation in the same way that that hairpin that Anbin Shui inserted into the hybrid globin CFOS globin constructs inhibited translation. And when intracellular iron concentrations are high, the cell will not want to bring more iron into the cell. It wants to reduce the amount of transferrin receptor. Therefore, it destabilizes transferrin receptor mRNA by there being no protective IREBP binding and nucleolytic endonucleolytic activity of the mRNA. And then the last example I want to talk about is how does histone mRNA get degraded at the end of S phase of the cell cycle? So it undergoes 3' terminal oligo-U addition by a TATase, a terminal U transferase. And this was one of a number of early studies showing how the addition of U's, unstructured U's, to an RNA can promote exosome-mediated decay. And you also know that U's can bind that LSM1 through 7 protein, which together with PAT recruit the decapping activity. And this is work from Bill Marsleff's lab in a really nice study where they took RNAs at the end of S phase of the cell cycle, decapped, so you get a 5' monophosphate, and circularized the RNA with T4 RNA ligase, and then used primers and cDNA and PCR to sequence across the ligated junction and determine what was that sequence. And it turns out that oligo-U can be added to the full length of the mRNA, as well as to regions that are upstream of that. And the idea is that the U's allow the exosome to enter into the unstructured region and degrade, and maybe there's a physical barrier, like there's the stem loop binding protein that would undoubtedly be a physical barrier to the exosome, which doesn't have great unwinding capability by itself. And so then the complex would fall off, and more U's would be added, and that would give the exosome kind of like a running start to get through some structured regions. So I think generally you'll find in the future that oligo-U addition to mRNA 3' ends will be used by a number of mRNAs to augment their decay by the exosome. And I'm done. So I'll take some questions.